Thank you, Anderson. And also many thanks to the Royal Swedish Academy of Sciences for this kind of invitation, and to the Crafoord Foundation for granting my project. And today I'm gonna talk about autoantibodies and autoreactive B cells protecting rheumatoid arthritis. So before the talk, I would like to disclose my competing interest. I'm mainly employed at Karolinska as a postdoc, and also I'm partially employed by Vakara, which is a small Swedish company dedicated in developing therapeutics against the rheumatoid arthritis. So rheumatoid arthritis is a kind of autoimmune disease which affects around 0.5% of the global population, and is driven by systemic autoimmunity. But the most predominant manifestation is the organ-specific inflammation, in this case synovitis. And its etiology is still incompletely understood, involving a complex interplay of genetic, environmental, and immunological factors. And all of our patients still require a lifelong management with mainly disease-modifying anti-rheumatic drugs. And until now, there's no cure proposed. So this is structure of a synovial joint with the two bone termini facing each other, and the interface is covered by cartilage with the synovium surrounding the articular compartment. And my work is mainly focused on cartilage. Here we could see that there are two layers composed of mostly collagens, proteoglycans, and the non-collagen proteins, with the collagen 2 being the most abundant component within cartilage. Over the past 2 decades, our lab has fine-mapped multiple epitopes on collagen 2, including one major T-cell epitope and several B-cell epitopes. The majority of the immune responses to these epitopes are pathogenic, driving arthritis progression, because if we clone antibodies from these epitopes and make a kind of a cocktail, inject them to the animals, we could induce synovitis. And if we use the most susceptible mouse strain, F6-2B here, we could induce arthritis within the time frame of 12 hours. So among all of the epitopes, the C1 epitope attracts much attention from us because we found that the antibody responses to this epitope are highly conserved between RA patients and animals immunized with collagen 2. And in this paper, we showed that anti-citrullinated protein antibodies could cause arthritis if they can cross-react to joint cartilage, especially the citrullinated C1 epitope. And then we cloned several antibodies targeting this epitope, like ACC1, C2C1, CB20, and most of them induce arthritis. So all this data suggests that the C1 specific B cells should exist, and indeed we found this population after enrichment across almost all species investigated, including mice, rats, and humans. And the interesting thing here is in RA patients, we could see that there was a significant reduction of this population. And in parallel, we observed that increase of C1 specific antibodies, indicating translation or a differentiation from B cells to plasma cells. But anyway, the presence of C1 specific B cells in the periphery suggests that they didn't undergo negative selection. So we then would like to know if they are positively selected. So first, we injected a germline-encoded antibody called CB20, which is specific for the C1 epitope, into animals, neonatal ones and adult mice. Here we found positive signals from especially neonatal mice close to the bone trabecula, indicating that the C1 epitope is exposed within the bone marrow. Then we thought if we knock in if we VDJ lock in the heavy chain of the CB20 antibody into this mouse, here denoted ACB mouse, they should have more C1 specific B cells and produce more C1 specific antibodies. Then they may develop spontaneous arthritis. But actually, this is the opposite. We found that the ACB mice are much more resistant to collagen-induced arthritis compared to the wild-type controls. And then we checked the signs of negative selection if they are affected by this procedure. But we didn't find any evidence. For instance, we subjected the C1 specific B cells from the ACB mice to mass spec, and we found that the light chain used by the C1 specific B cells is actually the same as the light chain of CB20, which suggests that they don't undergo the receptor editing. And then we made another mouse strain by mutating the key amino acid for CB20 binding. And then we back-crossed this strain to the ACB mouse. Therefore, they could not express the native C1 epitope. And then we found, regardless of their genotype of heterozygous or homozygous, they have these mutated mice have significantly lower numbers of C1 specific B cells compared to the ACB mice, indicating a possible positive selection here. Then it raises another question: why autoreactive B cells are positively selected? So to answer this, we performed such a transfer experiment by sorting ACB B cells and wild-type B cells, and then we transfer them to the recipient mice undergoing collagen-induced arthritis. But surprisingly, these C1 specific B cells suppress the inflammation potently. And this suppression is antigen specific because we didn't observe any difference in the MOG-induced EAE mouse model. Then we hypothesized the subset of B cells might be regulatory B cells, which inhibits inflammation through secreting IL-10. So we back-crossed the ACB mice to the IL-10 knockout background, but found that the suppression of arthritis is IL-10 independent. So they are not the so-called classic regulatory B cells either. Instead, we observed an expansion of Tregs in the ACB mice, and these Tregs were more activated and more proliferative. So to determine the interaction between C1 specific B cells and Tregs, we made such a system to co-culture them with or without direct contact. Here we showed that the C1 specific B cells can only expand Tregs when they have a direct contact. So this suggests that this subset of B cells is actually antigen-presenting cells, but not through secreting cytokines. Then we subjected those B cells to single-cell RNA sequencing, and we observed some elevated transcriptions of proteins such as CCR7 and CD72. Here we validated the findings by using flow cytometry, and the most interesting thing here is only these C1 specific B cells express CSR7 and CD72 at the same time, but not any other population. So because CSR7 is more relevant to cell migration, so we focused more on CD72, which also popped up from Christopher's data. We focused more on CD72 for its mechanism. Here we did 2 injections of anti-CD72 antibodies to the ACB mice. And we found that the suppressive function of these C1 specific B cells is largely impaired after the anti-CD72 treatment. Then we pre-treated these sorted C1 specific B cells in vitro and then transferred these pre-treated B cells into the recipient mice, and we found that its protective potential is almost lost compared to the cells treated with isotype control. So this may represent therapeutic target to select the antigen-specific B cells to prevent autoimmune diseases. And then when the autoimmunity progresses to plasma cell phase, we still find some protective mechanisms. For instance, we identified an epitope called F4 on collagen type 2. And we found that the antibody responses to this epitope are also negatively associated with RA activity. And our collaborators showed that antibodies to the citrinated F4 epitope have the same trend of negative association with arthritis. So we thought this could be a really good therapeutic target. Then we tried to make recombinant antibodies targeting this epitope by using phage display. Briefly, we expressed the F4 epitope on the surface of bacteriophages, and then we incubated these phages with 2 giant libraries containing up to 10 billion clones of single gene F3. And after the incubation, we eluted the bound clones and subjected them to sequencing, and they were denoted R69 series. And after a couple of rounds of selection, by checking their binding to synthesize the F4 epitope, recombinant F4 epitope, and cartilage tissue, we finally selected the one clone named R694, which demonstrates potent protection in both cartilage antibody-induced arthritis model and collagen-induced arthritis model. Then we phenotyped those animals and found that the neutrophil expansion was significantly restricted after the injection of R694 within both periphery and cellular fluid. Also, the recombinant F4 antibody also minimized the release of IL-1 beta into cellular fluid, which is critical for recruiting neutrophils. And then we supplement recombinant IL-1 beta to those mice pre-treated by the recombinant F4 antibody, but it failed to recover the arthritis development, indicating that this effect should be secondary to its protection. Then we focused more on the upstream pathway leading to IL-1 beta release. Here it has been shown that Fc gamma receptor 3 engagement is solely dependent on the release of IL-1 beta in antibody-induced arthritis model. So we measured the expression of this receptor, and interestingly, the expression was largely reduced, especially within the synovial fluid here. And this reduction occurred before neutrophil apoptosis because at an early time point, just 8 hours after the injection, the majority of the Fc gamma receptor 3 low population was Alex 5 negative, which suggests that they are not apoptotic, but with already downregulation of Fc gamma receptor 3. And this downregulation also affects the neutrophil capacity to phagocytose immune complexes, such as mouse IgG2b immune complexes. So here we checked how important this receptor is for arthritis development by injecting one antagonist specific for Fc gamma receptor 3 in Fc gamma receptor 2b knockout background, the 2.4 G2. So we could see here that the one dose of 2.4 G2 is sufficient to remit arthritis progression to a large extent. Then we wondered how it interacts with the Fc gamma receptors through the Fab or the Fc. To answer this, we mutated one amino acid which is critical for the binding to Fc gamma receptors and the position 297. Here denoted N297G. So the mutated version of the antibody almost lost its protection compared to the intact version of the antibody, suggesting the critical role of its Fc engagement. Then we also checked the Fab-mediated effect. From the immunoprecipitation, we actually identified a couple of targets, but in vivo, we only find the C1q subunits were significantly reduced within both serum and salome fluid. We also validated these results by cytometric bidary using many animals. So my current work is to investigate the mechanism of action of this antibody in our humanized mouse model, and this is also the project granted by the Crafoord Foundation. Our preliminary data showed that one dose of R694 could significantly reduce arthritis severity compared to the control. Here is glycine. So to summarize our findings, we basically discovered a subset of B cells specific for the C1 epitope, which could inhibit antigen-specific inflammation through activating and expanding Tregs. We also developed a subset of antibodies specific for the F4 epitope, which could bind to the F4 epitope for sure, and also some other proteins such as C1q. And this kind of binding could potentially deplete Fc gamma receptor 3 on neutrophils, therefore halting acute inflammation. Here we also cloned one ACPA called E4 from our patients, which represents the most common promiscuous ACPA found in our patients. And this clone also showed promising effect in experimental arthritis models. So reflecting on these discoveries, we proposed that there might be regulatory mechanisms at different stages in autoimmunity, such as protective B cells, collagen type 2 antibodies, and promiscuous ACPAs. And there is just a thin line between protective and pathogenic autoimmunity. So basically, we could probably develop therapeutics to manipulate this balance towards the protective spectrum. Here I would like to acknowledge my colleagues from the Beer Lab, especially my supervisor during my PhD, Professor Richard Hamdahl, and enormous gratitude to our collaborators from Zubrav Lab, Blom Lab, Fields Lab, Hamstrom Lab, and all the founding bodies. Thank you.